All righty, then we are going to talk about chapter 12, muscle physiology. So this may have to be split into two different videos because it could get long. So I'm going to start off. I'm going to see how far I get and keep the video to 45 minutes to 55 minutes. And if I go over that, then we'll go into two parts. So first of all, it's always good to review the organ system functions. So for skeletal muscle, most people get, produces movements. It pulls on bones, and the bones are really what move, but the muscles are causing that bone to move. Maintains your posture and body position, and also can support soft tissues, pelvic floor muscles, that kind of thing. Regulate the entering and exiting of material. That would be, hey, look, your urethral sphincter and your anal sphincter, those are skeletal muscles. Those are controlled by your somatic nervous system. You get to control those. That's a good thing. We don't want to be wearing diapers. So those are going to be skeletal muscle, even though the internal components or muscle leading up to those sphincters could be smooth muscle. Maintaining body temperature, this is partly due to the inefficient energy transfer system of cellular respiration and the inefficient energy transfer system from ATP molecules, the potential energy in that ATP, to the myosin head, and for the myosin head to then pivot, move, and slide the actin strand. And so since it's 100%, not 100% energy transfer, you're going to end up with some of that energy being lost in the form of heat. And that heat helps maintain your body temperature. Okay, so some anatomy review. Now remember, if you have not had anatomy and you want to review the anatomy prior, this is chapter 9 in the anatomy playlist. So chapter 9 in the anatomy will review all of the anatomy of the skeletal muscle. I am going to make the assumption that you have learned all this anatomy and I can just talk about the physiology. But that said, I will kind of break down that a muscle is really a series of compartments called fascicles that contain muscle fibers, and muscle fibers are the basic cells. So if you're looking at this is the cell of the muscle system. And so you have one muscle and it has an epimysium surrounding it. And then within that one muscle, you have bundles of muscle fibers or muscle cells. And those bundles of muscle fibers or muscle cells are referred to as muscle fascicles, bundles of cells. Each one has its own paramysium surrounding it. So this would be the paramysium. And then within this bundle of muscle cells, muscle fascicle, you're going to see a number of muscle fibers. So this is a muscle fiber, that's a muscle fiber, and so on. There are a number of muscle fibers in this muscle fascicle. Each muscle fiber is going to have its own connective sheet surrounding it called an endomysium. All of the endomysiums, all of the paramysiums, and the epimysium, they all come together, all those connective tissue sheets at the end, and form the tendon. So a tendon is really the coming together of all those connective tissue sheets of the multiple endomysiums, paramysium, and that one epimysium. So tendons are pretty strong structures and ensures the entire muscle works together as a unit. Now, if you take a muscle fiber, which is what we're going to be talking about mostly in physiology, that muscle fiber or cell is like any other cell, except it does have multiple nuclei to produce lots of proteins like enzymes and contractile proteins and so forth. So a lot of protein building going on, a lot of nuclei. There's going to be cytoplasm, but we don't call it cytoplasm. We call it sarcoplasm because sarco is with the muscle. And instead of cell membrane being a plasmalemma or a plasma membrane, we call it a sarcoplasm. So deep to the endomysium, you have the sarcolemma. Then you have sarcoplasm surrounding it. That's where glycolysis is going to occur. taking the energy out of the bonds of the glucose molecule and transferring it to ATP. If you want to then go into aerobic respiration, like we talked about, which is the Krebs cycle and oxidative phosphorylation, that's going to happen in these little guys right here, the mitochondria. So these are all mitochondria. Now you have these little red dots, and those red dots are called myofibrils. So these red dots are all myofibrils. And myofibrils contain sarcomeres. And sarcomeres are the smallest functional unit, meaning they actually do stuff in a muscle fiber. And so we're going to get down to the level of the myofibril and the sarcomeres in physiology. So just to recap, this chart's kind of nicely organizing a skeletal muscle. It's surrounded by a connective tissue sheet, epimysium, and it contains muscle fascicles. If you look at a muscle fascicle, it's surrounded by paramysiums and contains muscle fibers. If you look at a muscle fiber, it is surrounded by an endomysium and contains myofibrils. Now, this is the myofibril. This kind of is somewhat a representation, but this blue stuff here completely surrounds the myofibril. And so, yes, this is a myofibril, kind of a representation, but we'll look at what it looks like more realistically below. If you are talking about the smallest functional unit, that is this right here. This is a sarcomere. But as it's showing you, there is another sarcomere here that you just can't see, and another sarcomere here. And so basically myofibrils are sarcomeres attached to one another all the way down the myofibril. It's each sarcomere that's going to basically get shorter. Nothing's actually getting shorter, but we're going to talk about the red thin filaments sliding over the darker blue thick filaments. And so the illusion is that each sarcomere is getting shorter. And the actual is that all those sarcomeres attached to one another are all getting kind of shorter, and thus the myofibrils getting shorter, and all the myofibrils in the muscle are doing this. And so it looks like the muscle's getting shorter. But it's really sliding past one another. As we get down to it on a cellular level. So again, muscle fibers are multinucleated, right? Need all those nuclei to produce lots of proteins for not only to transfer energy from the food you ate to ATP so the muscles can slide and get shorter, but also to actively transport things like calcium and sodium potassium and generate action potentials. And so there's a lot going on. A lot of mitochondria to make sure you have adequate supply of ATP. And then we have these special structures. So we have a structure called a sarcoplasmic reticulum, and I'm going to start down here. It's a smooth endoplasmic reticulum, and it completely surrounds the myofibril. So each myofibril is going to have its own sarcoplasmic reticulum surrounding it. And there is then going to be what is called a T-tubule or a transverse tubule, which travels by each sarcoplasmic reticulum. And this is important because the sarcoplasmic reticulum stores calcium. And calcium is a signaling molecule to allow sarcomeres to start getting shorter, essentially, muscle contraction. And so there's an area around every myofibril where you're going to have the sarcoplasmic reticulum come in contact with the transverse tubule or T-tubule. And we're going to call that a triad. The lateral sacs or the terminal cisternae are here. They contain calcium in the sarcoplasmic reticulum. Now, The two-tubule is what the action potential or electrical current, how that travels throughout the muscle fiber. And so you have an electrical current traveling, and as you may already be guessing, as an electrical current travels through the T-tubule, it's going to open, guess what kind of channels? Voltage gated calcium channels, because we've seen this before. And this is going to be what allows calcium to leave the sarcoplasmic reticulum, come out, and then start becoming the signaling molecule for the muscle fiber to shorten. So we will get to that. So again, review sarcolemma, sarcoplasma, sarcoplasmic reticulum, transverse tubules, and the triads. We kind of went over this a little bit at the end of chapter 10. So we talked about the neuromuscular junction. And so we're also going to be reviewing this neuromuscular junction and how the release of acetylcholine at the neuromuscular junction can create an action potential that then travels down the T-tubules to release calcium from the sarcoplasmic reticulum and start a muscle shortening. So myofibrils, they are composed of sarcomeres attached end to end. So this is kind of showing you a muscle fiber. Remember, this is the cell. So a muscle can contain hundreds and thousands of muscle fibers. So we're just looking at one. Each muscle fiber has its own neuromuscular junction. So that is where the nervous system, the motor neuron, has chemical communication with the muscle fiber at the neuromuscular junction. And that's going to start generating action potential that then goes through the T-tubules, and everywhere that electrical impulse passes, it's going to open voltage-gated calcium channels so calcium can leave the sarcoplasmic reticulum and go out into the sarcoplasm surrounding the myofibrils. Sarcomeres, again, this is showing you, here's a sarcomere, there's a sarcomere, there's a sarcomere. They're attached all the way down the myofibrils. Sarcomeres are the unit that actually moves. And so each sarcomere is moving. That's why it's the smallest functional unit. Looking at the structure microscopically of a sarcomere on a myofibril, we just look at one. We have thick and thin filaments. Now, the real name for a thick filament is myosin, and thin filament is actin. So we need to remember that first. Actin is the thin filament, myosin is the thick filament. And so if you're looking at the dots, the myosin are the big yellowish dots, and actin are the smaller blue dots. And so if you're looking at this cross section here, you can see that the thick filaments have lots of little filaments, thin filaments surrounding it. And this is because these thick filaments have little heads that they can reach over and attach to the thin filaments and pull those thin filaments and slide those thin filaments over. And so this one myofibril, you can see there's going to be a lot of opportunity for the myosin heads to reach over and grab a bunch of thin filaments. And so when we're looking at it just from this perspective, you don't realize that in 3 dimensions, it's a lot more than just what you're seeing in a flat surface for making connections. And when a thick filament reaches over and grabs onto a thin filament, that is referred to as a cross bridge. So a lot of opportunities for a lot of cross bridges. Now, I'm talking about thick and thin filaments. So let's go even a little deeper into that. And we have the thin filament. So the thin filament, again, is actin. I should probably draw in another color so you can see a little bit better. So actin. Actin is the thin filament, and on this thin filament, there are a couple of regulatory proteins. So let's start with the troponin complex. So this, and let me get a brighter color for you guys. This right here, and that is not very bright, is it? Let's try this. This right here is a troponin complex. This right here is a troponin complex. Now, what a troponin complex is for is to bind calcium. So when calcium is let out of the sarcoplasmic reticulum, that calcium can bind to troponin. And when a calcium binds to troponin, say calcium binds on that troponin, it's going to move this regulatory protein strand here and here. It's going to move it off these little black dots. And what are these little black dots here? These are the active sites for myosin heads to attach to. So when calcium is released from the sarcoplasmic reticulum, then those voltage-gated calcium channels in the sarcoplasmic reticulum open because an electrical action potential is a change in voltage and opens those voltage-gated channels. Calcium's let out and it can bind to troponin. It will then move tropomyosin off the active sites. And will expose these active sites. Then these myosin head can reach over and attach to the actin strand. If tropomyosin is over the active sites like here, the myosin head can't bind. Doesn't happen. But if you see here, when calcium binds to the troponin complex, it moves tropomyosin off these active sites here. And then the myosin head can reach over and attach. And there might be a myosin head reaching over and attaching here, and another one here, and another one here. The point is, if you have active sites available, myosin heads can now bind to the actin strand. Okay, so myosin, this is the thick filament. And these are called myofilaments. And this is the thin filament, or actin, also a myofilament. So a couple of things when we're looking at a sarcomere. We're looking at actin, thin filament, myosin, thick filament. Actin, thin filament is blue. And then again, myosin, the thick. So you can see these little guys right here. Those are the myosin heads sticking up. So actin and myosin are contractile proteins. On the actin strand, that is the thin strand, it's going to have the troponin complex to bind calcium and tropomyosin, which is a regulatory protein that covers up active sites when you don't want your muscle to form cross bridges and move. And so I do need you to make sure you understand that actin's thin filament, and it has tropomyosin and troponin. Troponin has a place for calcium to bind, and when it binds to the troponin complex, it moves tropomyosin off the active sites. Thick filament, myosin. Myosin has these long strands with heads, and the heads are towards the actin strand, which is part of that I band, and they are the little things that can pivot. So a couple of things about these myosin heads. They have enzymes on them, and those enzymes are ATPase enzymes. And what this enzyme does here, is it is going to essentially take the potential energy that's stored in ATP and it's going to take that energy, that's potential energy that's stored in ATP, and transfer that energy into the myosin head. So now that myosin head is energized, and that's the energy that's going to allow the myosin head to pivot once it attaches to the actin strand, and it's going to move that actin strand. And so that's actually where the sliding or movement is coming in a sarcomere, but it's happening in every sarcomere all the way down the myofibril and in every myofibril in that muscle fiber and every muscle fiber in that motor unit, and so on and so forth. So you can see these myosin heads basically have, or these myosin have two heads, and the way this thick filament, or what we call myosin, is when it's put together, it's a bunch of these myosin strands put together. And so you have a lot of myosin heads going in a lot of different directions that can attach to a lot of actin strands. So think back to this picture, right? You're seeing it here. If all these are myosin, they're all going to have heads sticking up in all the different directions, and they can glom onto all those different actin strands in 136 degrees. Okay, 360 degrees. Now I'm just babbling. So that's a thick strand. So when we put them together, oh, and let me mention one thing, by the way. This ATPase enzyme on the myosin head, it's either going to be really fast enzymatic action, in which case it can not only take the potential energy out of ATP quickly, but it can also make that myosin head pivot quickly. That's what we're going to call a fast twitch muscle fiber. Fast twitch muscle fibers have fast ATPase activity and can move fast. So fast twitch equals fast ATPase on myosin head. So fast twitch muscle fibers have fast ATPase activity and can move the myosin heads and have them pivot really quick, detach and reattach and pivot quick. Those are going to be fast twitch. Slow twitch, slower ATPase activity, but doesn't use ATP as quickly, tends to have more capillary oxygen infrastructure and create energy as it's being used. So those are the slower twitch muscle fibers, and we talk about that later. Putting it together with the sarcomere. So you have the actin thin strands, and you have the myosin thick strands. And this is a sarcomere from what is called Z line, kind of zigzag to Z line. And from that Z line, it's going to become another sarcomere, right? So this will be another sarcomere here. Each sarcomere, if calcium is let out and binds, is going to have the ability to cross bridge. And when it cross bridges, those myosin heads are going to pivot towards the midline. And what that does is it slides the actin strands this way. Myosin's not moving. Actin strands are being pulled over the myosin. And when they attach, that's called a cross bridge. So it says cross bridge down here. It's only a cross bridge if that myosin head actually attaches here to the actin strand. I go right here. That would be a cross bridge. Now there's tentin, this elastic fibers that connect to the myosin strand, and it allows the basically the sarcomere to return to resting length, kind of pull it back, a little elastic component to it, and so muscles can return to their original length. So now I'm going to kind of put it all together. So we talk about sliding filament theory. Well, I have been talking about it. Myosin heads pull the actin strands and slide them over the myosin strands. So nothing shortening, but things are sliding over one another. And so the effect is, yes, the sarcomere does get less wide, shorter, should we say, but it's really filaments sliding over one another. And how that happens is myosin heads attach to the actin strand, form a cross bridge, pivot, pull that actin strand, and then the myosin head detaches and then can re-energize, reattach, and pivot again. That's called the cross bridge cycle. And what gets that all going is the interplay between the nervous system and the muscle fibers, and that's the excitation contraction coupling. And towards the end, we're going to talk about different muscle cells and how they generate ATP and how they use ATP in the cross bridge cycle. So starting with the sliding filament theory, again, the muscle is, quote, shortening, but no structures are getting shorter. It's filaments sliding past one another. We know this because we can stain muscle fibers down to the sarcomere. And if you're looking at this sarcomere, that right there is going to be a Z line. So that's a Z line. And that is a Z line. Now, this is at rest. If the muscle, quote, contracts, and you look at the sarcomere, what you see is this band right here, the A band, that is myosin. And the myosin does not change in width. Myosin stays the same. But where you have actin, the I bands, that gets significantly smaller. And so what we're seeing here is that the actin are getting pulled over the myosin. And so when there was only actin at rest, you see a lot of the strand, but when it's fully contracted, you see that it then blends in with the A band, which is the myosin band. So we know that these actin strands are getting pulled and sliding over the myosin. The Z lines are getting closer together, but it's really filaments sliding past one another. And so the sliding filament theory is that if the myosin heads can attach, form cross bridges with the actin strand, they're going to pull the actin strand closer to the midline and slide those actin strands over the myosin, which makes it closer to the midline. Now, if we're going to add the cross bridge cycle, we're going to talk about those myosin heads and how they slide the actin strand over the myosin strand. Here's the kind of beginning of it, right? So we're going to start with calcium is released from the sarcoplasmic reticulum. Why? Because at the neuromuscular junction, the motor neuron released acetylcholine onto the motor end plate of the muscle fiber where the receptors, the nicotinic cholinergic receptors, are there to bind the acetylcholine. That is an ionotropic cholinergic receptor, which means when the acetylcholine binds to the receptor, it opens up and allows sodium into the muscle cell. That is going to depolarize the muscle cell, generating an action potential or electrical current that then travels down the T-tubules of that muscle fiber. And at every triad where the T-tubule passes by the sarcoplasmic reticulum terminal cisternae, it's going to open up voltage-gated calcium channels and allow calcium out. So that is where we get to this place here, where the calcium begins to rise. More frequency of action potentials sent to the neuromuscular junction, the more frequent action potentials are generated in the muscle fiber, the more the voltage-gated calcium channels stay open, and more calcium is released out into the sarcoplasm. That allows for more calcium to then bind to the troponin complex. This then moves tropomyosin off, and now you have exposed binding sites or active sites for myosin heads to bind. Now, I kind of got ahead of myself because I forgot to mention that you need to energize those myosin heads. That is that enzymatic, you have an enzyme that takes the energy out of ATP by separating that high inorganic energy phosphate bond and puts that energy into the myosin head. It can attach, it's energized. So it's the binding of an energized myosin head, and that forms a cross bridge. So right here, we're looking at a cross bridge. I always kind of shorten it to cross bridge. So now you have a cross bridge. The minute that cross bridge happens, it's what is called a power stroke. The power stroke is the myosin head pivoting and pulling the actin strand with it. You have the power stroke, actin strands pulled to slide over the myosin. You have a new ATP bind to the myosin head. Now ATP is bound to the myosin head. That changes the conformation of the myosin head, and it detaches from the active site on the actin strand. And then it will re-energize myosin head. Right, so the ATPase hydrolyzes or takes out the energy from the ATP high energy phosphate bond and puts it in the myosin head. That's a re-energized myosin head. Now that myosin head is re-energized and can start that cycle over. And so thus it becomes a cross bridge cycle. Cyclical in nature until one of two things happens. As soon as there's no electrical impulses passing through the T tubules, calcium the whole time is getting actively transported back into the sarcoplasmic reticulum. So calcium might be let out when voltage gated calcium channels open, when a action potential goes past that sarcoplasmic reticulum, but it's immediately being pumped actively, energy, back into the sarcoplasmic reticulum. That's so you can start and stop your muscle fibers quickly. So you have to have a steady stream of action potentials going to that muscle fiber in order to keep calcium out so you can continue to cross bridge. So calcium needs to be present for the cross bridge cycle to continue, as well as ATP production needs to continue. There has to be new ATP. First of all, you need ATP to energize the myosin head and actually have it cross bridge and pivot to a power stroke. Second, you need another ATP to re-energize and detach that myosin head. So you need another ATP to release the myosin head from the actin strand. And that essentially is what you see in rigor mortis, which is, or rigor, it's where you don't have an additional ATP to release the myosin head, and so it stays attached to the actin strand, and that muscle stays in the same configuration as when the person had passed away or ceased to produce ATP, ceased cellular respiration. And so reading through these, understand it kind of is step one. And I want you to do this yourself, really, because it's the only way for you to put this into context you understand, is you do your first box, right? ATP hydrolyzes, right? Number one, hydrolyzes the myosin head and creates an energized myosin head. And if there's calcium present on troponin, tropomyosin will expose active sites in the actin strand for that myosin head to bind. That will then form a cross bridge. The binding of the myosin head to the actin releases the energy in the stored myosin head, and it will cause a power stroke, or the myosin head to pivot and pull the actin strand, power stroke, pull that actin strand closer to the midline. You have to then have a new ATP or second ATP bind to the myosin head to decrease the affinity of that myosin head for the actin strand, i.e. myosin head releases actin strand, and then that myosin head gets re-energized and can start the cycle all over again. So that's the cross bridge cycle. Now, again, a power stroke is when the myosin head attaches to the actin strand and releases the energy stored in that myosin head, thus pulling or propelling the actin strand closer to the midline. So one thing to take into account is that you can go through these. You can pause me right now and go through each one of these, because I am going to fly through these very quickly. But you have the option of pausing or slowing me down. So number one, you have the enzymatic activity, or the ATPase, on the myosin head. It's going to bind an ATP molecule, hydrolyze it. That means separate that inorganic phosphate from adenosine diphosphate. And that's where the energy is stored. And it's going to put that stored energy into the myosin head. And then it can bind to the actin strand if calcium is bound to troponin and exposes the active sites, because tropomyosin moved off, right? So that's tropomyosin. Inorganic phosphate releases, energy gets released in the myosin head, causes a power stroke. Actin gets pulled towards the middle of the sarcomere. After actin gets pulled towards the middle of the sarcomere, the myosin head releases adenosine diphosphate. Remember, it already released the inorganic phosphate. ATP means triphosphate. ADP is 2, and there's the third. Now, in the low energy form, ATP, a new ATP has to bind to the myosin head to then lower the affinity for the actin strand. So the myosin head then releases the actin strand here, or unbinding of the myosin. That new ATP is hydrolyzed by the ATPase enzyme, re-energized, and can now form a new cross bridge. And so that's the cross bridge cycle. Keep in mind, ATP is being used in the muscle fiber really in 3 distinct ways. One, to energize the myosin head so it can bind to the actin strand, and then have the energy in the myosin strand pivot and pull the actin strand. 2 is another ATP has to bind to the myosin head to release that myosin head from the actin strand, i.e. lower the affinity for the myosin head to the actin strand and have it released. The third way is going to be active transport of calcium back into the sarcoplasmic reticulum. So muscle fibers are definitely high ATP users, high energy users, and thus require a constant supply of energy. And we'll talk about that when we talk about how each muscle gets to have its own storage of glucose in the form, and storage form is called glycogen. So each muscle has its own little cubby or storage of glycogen for energy, and the availability of triglycerides, lipids to be used as energy, and a lot of blood supply because they are energy consumers. Now, we're going to add in the neuromuscular junction, which is where the nervous system then starts and stops the muscle from doing the cross bridge cycle. So we're kind of putting it all together. So each muscle fiber has a neuromuscular junction. And a neuromuscular junction is where the motor neuron axon comes in contact with the skeletal muscle fiber, and it's like the on-off switch. But a motor neuron can actually innervate several muscle fibers or cells. So what happens is you have a motor neuron coming down, and it can branch and have a number of terminals make contact with different muscle fibers, right? So if this was a motor neuron, it can branch and innervate muscle fiber 1, 2, 3, 4, and this would be actually called a motor unit. So we talk about that again a little bit later. But that's what's meant by each motor neuron can innervate several muscle fibers. If this motor neuron has an action potential, all of these then form action potentials at the same time. So if the motor neuron activates, all the muscle fibers that innervates in that motor unit are going to activate at the same time. Now, this is the motor neuron, synaptic terminal. And on the muscle fiber, so this right here is the muscle fiber, the sarcolemma or cell membrane, the area where it folds in and makes contact with the axon terminal is called the motor end plate. So the motor end plate on a muscle fiber has the cholinergic nicotinic receptors or acetylcholine. So these are those receptors right here. They're on the motor end plate of the muscle fiber. And same thing, right? Action potential comes down the synaptic terminal, causes voltage-gated calcium channels to open. Calcium comes in, causes the exocytosis of neurotransmitter, and that neurotransmitter is acetylcholine. Acetylcholine on a nicotinic cholinergic receptor is always excitatory, meaning it's always going to let sodium in, and an action potential is generated. Right? Sodium comes in, new action potential is generated, and that action potential is going to travel down all of the transverse tubules or T-tubules. Now, every place or every triad that action potential goes past, it's then going to open voltage-gated calcium channels, and then calcium channels are let out. And that's what trigger the release of calcium from the sarcoplasmic reticulum. Calcium's in the environment, binds to troponin complexes, moves tropomyosin, opens up these active sites for cross-bridging. And so now we're coupling excitation at the neuromuscular junction here with the cross-bridge cycle that's going on in every single sarcomere and every myofibril in the entire muscle fiber. So going through this, the motor end plate on the sarcolemma has a high density of nicotinic cholinergic receptors for acetylcholine. It's always going to be excitatory. It's always going to cause the muscle to generate an action potential. So calcium signaling molecule stored in the sarcoplasmic reticulum binds to troponin, causes tropomyosin to move off active sites, or myosin heads to bind. If there's no calcium, tropomyosin stays over the binding sites, no cross bridge. Muscle cannot contract. But if calcium is present, if it's released from the sarcoplasmic reticulum, it's going to ultimately bind to troponin, move tropomyosin, and expose binding sites for myosin and cross bridges to form. Now, we've spent a lot of time in this class talking about concentrations, right? And the higher the concentration of calcium, the more calcium is going to be bound to troponin. The more troponin molecules that bind calcium, the more exposed binding sites for myosin. Thus, the more cross bridges can form. So you can say more action potentials equal more calcium release from the sarcoplasmic reticulum, more calcium troponin, more exposed active sites, more cross bridges formed. And you're like, okay, cool. What does that mean? That means, I'm going to do this big and bold. What does that mean? More force produced in muscle fiber. So it all starts here. More action potentials. If you release more calcium, you have higher concentration of calcium to bind to troponin. That's going to equal more exposed active sites, which then results in more cross bridges. And the number of cross bridges that are formed are going to equate to more force in the muscle fiber. So frequency of action potential is going to ultimately result in more force production. So again, going through this, steps of excitation, contraction, coupling. Action potential in a sarcolemma travels down the transverse or T tubules, opens up voltage-gated calcium channels in the sarcoplasmic reticulum and increases the amount of calcium in the cytosol. That's going to increase the likelihood that more troponin is bound with calcium, which is going to increase the active sites available for cross-bridging. And so again, this goes through voltage-gated calcium channels, calcium release, calcium binding, cross bridges. But remember, calcium is being actively transported back into the sarcoplasmic reticulum. So you have to maintain action potentials to maintain calcium out, to maintain cross bridging. And so that's how the nervous system can also stop muscle contractions. If there's no more calcium because it's actively transported back, no more active sites exposed, tropomyosin blocks the active sites, no more cross-bridging. And so again, different ways you can do this. I have this where, I did this myself just to kind of show students. It's good to write this down, what's happening, right? Upper motor neuron is sent from the primary motor cortex in the central nervous system, and it leaves on a lower motor neuron in the spinal cord out to the peripheral nervous system, which is your somatic nervous system, skeletal muscle. And that's going to ultimately release acetylcholine at the neuromuscular junction. That acetylcholine binds to receptors on the motor end plate. They are nicotinic cholinergic receptors, which mean they open ion channels for sodium to flood in and create an action potential that will then travel down the T tubules, transverse tubules to release calcium through the sarcoplasmic reticulum that it comes in contact with, and so on, start the cross bridge cycle. And again, this will continue. So a couple more things, and I think I'm gonna end it because it's getting to be late. But I want to kind of review the fact that muscles produce force, right? And that force is generally to overcome a load. A load is whatever your muscle is trying to move. So it's the force opposing the muscle. If the muscle stays the same length, it's called isometric. Iso means same, metric is length. If the load stays the same and the muscle's moving, it's isotonic. So there's isometric contractions and isotonic contractions that we're going to talk about. There's also isokinetic, but I'm not talking about those. What's actually moving are the sarcomeres. And each sarcomere is sliding and pulling on each other, which makes the whole myofibril kind of get smaller, right? Which then all the myofibrils in the muscle fiber get smaller, and then all the muscle fibers that are contracting are getting smaller, and they're pulling on the tendons. And those tendons are going to move something or just keep it the same. Basic biomechanics are this. So this is kind of showing you a muscle. And you have two tendons. You guys kind of know this, right? So you have a tendon here and a tendon there. You have an origin and the insertion. And insertions move, origins don't generally move. And when we talk about biomechanical advantages and how much force does it take this muscle? So you have a muscle here and we're going to say, you know, how much force does this muscle need to produce to pull here at the tendon site to move this weight, right? Because this 10 kilograms is pulling down. So this is the load. And this muscle has to produce force. And that force being produced in the muscle is going up on this plane, and it's pulling from the insertion, which is here on the radius. Now, the focal point, a fulcrum, is the elbow, because it's a hinge joint, because it does flexion and extension. And you want to move this 10 kilogram up into flexion. We don't know how much force. We don't know F. What does the muscle have to do? We know it has to move 10 kilograms of a load, but we don't know how much force. What's the force? This is a very simple biomechanical, but what we're looking at here is you look at where the insertion is and the distance from the fulcrum, because that's where this muscle is pulling from, the insertion. And then, so that is going to be, we don't know force, but we know that the insertion is 5 centimeters away from the fulcrum. We do know the load, right? The load's 10 kilograms. And we also know that it's actually 35 centimeters away from the fulcrum. And so what's the force? How do we figure that out? Oops. Is you basically go 5 F, and that's 350, and you divide by 5, and the force needed is going to be 70 kilograms. So the muscle has to produce 70 kilograms of force to overcome 10 kilogram load. Now, that's with a tendon insertion that's 5 centimeters away from the fulcrum. If you say have an insertion here that is 10 centimeters away, right? So the insertion is a little bit farther or more distal on the radius. If you do that same math, it's F10 equals 350, you divide by 10, now that muscle only needs to produce 35 kilograms of force. So that is to say, the muscle insertion and where it is located and how far it's located from the fulcrum determines how much force the muscle has to produce. And so biomechanically, some people can have a biomechanical advantage, meaning they can produce less force in a muscle to move the same amount of weight if their insertion is, say, farther away from the elbow or olecranon. And that's generally the difference between males and females. Males tend to have an insertion, a bicep brachii insertion farther or more distal on the radius than a female insertion. And so male biceps brachii can move the same amount of load with less force needed in the muscle. So that's an example of a biomechanical advantage. As far as twitches, there are 3 phases that a muscle goes through, right? Tension in a muscle fiber. And the first phase is called the latent phase. And the latent phase is the period of time from the action potential at the neuromuscular junction to the onset of the contraction or actually moving. It's where calcium is being released from the sarcoplasmic reticulum, but not enough tension has been produced yet to make the muscle move. And when the movement starts, then it becomes the contraction phase. And then when the movement stops and the muscle goes back to resting muscle tension, that is the relaxation phase. Now, in an isometric twitch contraction, the load is generally greater than the tension. Therefore, the muscle is not moving the load so nothing's moving. Isotonic, the tension in the muscle is greater than the load, so the load or weight or whatever is moving. It's important to understand that enough cross bridges have to form, enough myosin heads have to attach to the actin strand to overcome a load. And so another way to think of the latent period is that it's the period of time it takes your muscle fiber to create enough cross bridges, enough myosin heads, pulling on enough actin strands at the same time to produce enough force to overcome whatever load you're trying to ask that muscle to move. And so when you look at the time it takes to actually contract, which is distance shortened, if you have a heavy load, what you're going to notice is if you have an action potential here and the load is heavy, which is this purple, it's going to take time to build up cross bridges to start moving the load. And you're not going to be able to move it very far because you're probably using all your cross bridges and all those myosin heads to move that load. Now, if you have a slightly less weight, right, then you have to have enough cross bridges to move an intermediate weight. Well, that's less cross bridges, so less cross bridges take less time, and you could probably move it farther. And then light load, you don't have to form many cross bridges at all, so it can happen fast and go through a lot of movement. And so that's the first thing. Second thing we have to take into account, fast twitch, slow twitch. So again, myosin heads can have fast ATPase, which means it's going to be able to form cross bridges faster and activate and pull those myosin faster. So that's going to make a difference in this scenario when you're looking at a whole muscle. And there's also the calcium being pumped back into the sarcoplasmic reticulum can also be fast or slow. So calcium gets pumped back into the sarcoplasmic reticulum faster in a fast twitch muscle fiber than in a slow twitch muscle fiber. And then also, just those two combined are going to be factors in the speed at which a muscle fiber can overcome a load. So kind of just to summarize, muscles are graded, really have graded contractions, right? So first you want to think about it is the tension produced in an individual muscle fiber in those motor units, right? So one motor unit and how much tension it's producing in the muscle fibers it innervates. And that comes down to the number of cross bridges formed. And what determines the number of cross bridges formed? The frequency of stimulation of that motor neuron. Because the frequency of stimulation of the motor neuron determines how many voltage-gated calcium channels open, and thus how much calcium is released. And how much calcium is released determines how many cross bridges can be formed. And then not all motor units are the same, right? So some motor units are really large motor units, meaning one motor neuron innervates hundreds of muscle fibers. So if you have a high frequency of stimulation of a large motor unit, you're going to have a lot of muscle fibers doing the same thing at the same time. And so that's going to bring a bigger force in that muscle. And then you can start increasing the number of motor units activated to even create more force or tension in that muscle, right? So muscles can start at the level of the skeletal muscle fibers in a motor unit by the frequency of action potentials arriving at the neuromuscular junction of all the muscle fibers innervated by that motor neuron. And then it can start activating more motor neurons and more motor units. And so you have an ability for muscle fibers to generate force on a graded or gradated basis, right? And so that's where you start getting into the principle of recruitment. So we're going to end here, and then we're going to start talking about recruitment and force velocity relationships. And we'll have a second video on that. So stay tuned.